In a world beneath our own, humans are no longer humans and creatures are not the ones we are familiar with. The world is becoming a technological center rather than a natural one, and humans will no longer have to rely on the limitations they were born with. Is that something positive or bad? From the intricacies of biotechnology, organ farms, and artificial wombs, we are continually searching for ways to improve the world around us. Whether or not this is harmful to our environment, the short-term benefits appear to be on our side, which could blind us to the long-term damage that is approaching. Join me as I reveal the concealed complexities of this world and how they will affect us and future generations. Hello everyone, welcome back to Zeey. Subscribe to our channel and ring the bell so you won't miss any update. Bioprinting, the mysterious coupling of biology and technology. Who has not yet attempted to 3D print a miniature structure or an ordinary cup? My recommendation, it is a must-try and so incredible. Now that is not all that 3D printing can do. In fact, people have attempted to build real structures and houses with 3D printers, which is simply amazing. However, there is always at least one individual who goes a bit too far and suggests 3D printing humans. Yes, that is correct. You heard me correctly. And it's about to get a little spooky, so don't view this video in the dark and make sure you're well protected. This technique is known as bioprinting, which is very similar to the concept of 3D printing, but instead of using plastics or metals, they use cells and biomaterials to construct structures that function almost like living organs by causing the cells to divide and multiply. Steps are involved in bioprinting. Let's examine how they work together. Initially, there's pre-bioprinting. This elaborate term simply refers to the process of creating a printer-friendly file. Consider it similar to creating a recipe for the printer to follow. Using complex examinations such as CT and MRI, scientists ensure they have all the necessary components. They combine cells with a special ink and use a specialized camera system to ensure that they have sufficient cells to produce a tissue model. Following is bioprinting. Here's where the true miracle occurs. Scientists place a pigment-containing cells in a cartridge. They then select the appropriate tools, similar to selecting different paintbrushes for various art endeavors. Scientists must use various cells, inks, and equipment depending on the type of tissue they are attempting to construct. It's comparable to creating a miniature work of art with living cells. Finally, there's post-bioprinting. Here the printed structures receive their final details. Similar to allowing a cake to chill or a painting to dry, the structures must become stable. This is accomplished by soaking them in a special solution or bombarding them with ultraviolet radiation. Based on the composition of the structures, scientists determine which technique to use. The structures are then placed in an incubator to grow and develop. It's similar to giving them a warm and comfortable place to chill out while they transform into actual tissues. This is extremely beneficial for contemporary medicine and treatments. Your eye no longer functions as it once did, or has your hair ceased to grow uniformly on your head? Well, scientists have the biological solution, so you don't need to contact 70 hair transplant specialists. Biotechnology will open new avenues for more effective wound healing after life-altering surgeries and far beyond that, as far as your imagination will carry you. Organ farms. This bioprinting technique is not applicable everywhere. It requires a very specific and distinct environment in order to be more effective. These environments are known as microgravity environments, and as their name implies, they are places where gravity is extremely low. I'm sure you're wondering why, so here's the rationale. Creating the vascular networks required for organ function can be a challenging endeavor. The majority of bioinks or hydrogels are liquid at ambient temperature but solidify when heated to body temperature. This implies that bioprinted structures require support to prevent collapse. However, using scaffolds as support can be detrimental to the vascular networks. To circumvent these issues, scientists are investigating bioprinting in a zero-gravity environment. They anticipate that this will allow organs to grow without the need for scaffolds. This could be the key to bioprinting organs that are completely functional. In May of 2019, NASA sent a biofabrication facility to the International Space Station to test this theory. Their goal is to discover a solution to the organ shortage, which results in an average of 20 deaths per day in the United States among those waiting for transplants. Why delay if you can produce? The group began by completing test protocols and aims to begin printing entire organs by 2025. It may take an additional decade before these organs can be utilized in clinical settings. Despite the high cost of rocket missions, it is anticipated that the cost of NASA organs will be less than what patients presently pay to treat chronic diseases. When you gaze up at the International Space Station flying just above you, you should now realize that in addition to carrying humans to explore the Earth and beyond, it also carries potential human organs. Would you like to have bionic eyes that can see in all wavelengths and can telescope in or out as required? As cool as that may be, I believe it could be overwhelming for the average person's daily existence. Sometimes not possessing everything is sufficient for our mental well-being. What is your opinion? Let me know in the comments. Let's have a discussion. Artificial wombs. Babies conceived without sperm or eggs. How? Well, as I've previously stated, don't let the limitations of your imagination convince you that we cannot accomplish the inconceivable. In recent years, significant advances have been made in the field of reproductive science, challenging the conventional conception of conception. Using revolutionary technologies and cutting-edge techniques, scientists are exploring alternative methods to produce offspring without sperm and eggs. In vitro gametogenesis, IVG, is an example of such a method. IVG is the process of creating gametes or reproductive cells from epidermis or blood cells. These cells are transformed into either sperm or egg cells through a series of laboratory manipulations. Obtaining a small sample of the individual cells, typically epidermis or blood cells, is the initial step. Then, using induced pluripotent stem cells, researchers reprogram these cells to their embryonic state. This enables their transformation into any other cell type, including the germ cells that produce sperm and ova. The reprogrammed cells are then exposed to a carefully orchestrated sequence of chemical signals, which fosters their maturation into fully functional gametes. Scientists closely monitor this process to ensure that the cells undergo the required maturation phases to become viable reproductive cells. Once the gametes have been developed effectively, they can be used for fertilization. In the case of sperm cell synthesis through biotechnology, these synthetic gametes can be used for in vitro fertilization, IVF, to fertilize an egg. Similarly, conventional sperm can fertilize artificial embryos. IVG's capabilities are not restricted to reproduction without conventional gametes. It also provides opportunities for same-sex couples and individuals unable to produce viable ova or sperm. For instance, two men's skin cells could be reprogrammed into ova, allowing them to have biological children together. Similarly, two women could have their cells transformed into sperm to enable conception via IVF. There are a number of ethical considerations associated with this innovative technology, despite the fact that its potential is undeniably vast. Concerns regarding the ramifications of modifying human reproduction and the potential dangers of manipulating cells must be carefully addressed and investigated. However, as science continues to press the boundaries of what was once considered impossible, the possibility of babies conceived without conventional sperm or eggs illuminates a future where reproductive options are no longer limited and genes are no longer predetermined. The ecosystem. These developments are both beneficial and detrimental to the environment. It depends on how we utilize the advancement of technology while taking into account the surrounding environment. Due to rising global temperatures, living near the water may become hazardous. This is damaging locations such as Australia's Great Barrier Reef. However, there is a possible remedy. Scientists are utilizing synthetic biology to make coral more robust and tolerant of warming water. We wanted to find out what people believe about genetically modified coral, so we surveyed over 1,000 Australians. It turns out that more individuals support the concept than oppose it. However, many remain uncertain and wish to ensure that we are doing the correct thing. They are also interested in learning more about the potential repercussions and believe we should use this technology with caution. People are optimistic that this could save the reef and provide some benefits, but they want additional testing and regulations before diving in. What are your thoughts on this solution? Is it more or less detrimental than advantageous? But it goes even further than that, as scientists who have hacked the functions of nature are investigating ways to give animals their own interest-based functions, drones in the form of birds, new tools for espionage and global occupation. Will the newly created animals be able to coexist with their natural counterparts, or will war ensue? The future will tell. New species? Few appear to be bothered by the fact that humans have many limitations. What is the solution? Surely you predicted correctly, biotechnology. Bioluminescence is a fantastic method to produce non-harmful light naturally, and biohackers are on the verge, if not already able to glow in the dark and produce light. They have also discovered methods to produce non-perishable, allergen, and disease-free foods. Therefore, humans are essentially becoming superpowered machines. Cryosleep, mind-blowing cerebral enhancements, and even AI fusion. It is as if we are producing an entirely new species. However, there is a caveat. We might lose our compassion as a result. Indeed, becoming emotionless machines is a valid fear. No one can predict where this will lead us as the distinction between humans and robots continues to dissolve. And what about justice? Will only the wealthy receive these interesting upgrades? We can't have a greater disparity between the wealthy and the poor, right? We require rules and regulations to maintain order. The future is both amazing and frightening. Using biotechnology, we can perform astounding feats and solve a variety of issues. However, we could also make a muddle of things and lose what makes us human. Therefore, it is up to us to act intelligently. Let's deliberate it out and determine the best course of action. We cannot allow the human era to become a catastrophe. We must discover a compromise between progress and ethics. It is possible, but we must strive for it. And there you have it, guys, the terrifying conclusion of the human age. I hope you enjoyed this whirlwind tour of the world of biotechnology and its potential repercussions. Remember, although the future may be extraordinary, it is our responsibility to approach it with intelligence and integrity. Let us not sacrifice our compassion in the name of progress. Until next time, remain human and continue to enjoy. Thank you for watching, and we'll see you in the next video, in which we'll discuss more fascinating global developments. Don't neglect to like and subscribe to our channel to receive updates on our newest videos and most exciting topics.